In this lecture, we're going to review a process called excitation-contraction coupling. And this is referring to two processes that will come together that provides the link between action potential generation and force production inside skeletal muscle. And all this is centered on the release of calcium from stores inside the muscle cell, and of course those stores refers to the sarcoplasmic reticulum. So this is what's often called electromechanical coupling, and the electromechanical coupling is a communication between a voltage sensor, which refers to a protein embedded deep inside the transverse tubule, and a calcium release channel. Now this calcium release channel is a protein that will allow calcium to move from stores inside the sarcoplasmic reticulum, will not leave the cell, but will migrate and disperse within the cytoplasm of that cell. The opening of that calcium release channel has to come from activation of another protein called a voltage sensor, and that voltage it is sensing is the action potential that was originally generated from the connection between a terminal axon and that skeletal muscle fiber. The voltage sensor is specifically referred to as the DHP receptor that we'll touch on here in the next couple of slides. The calcium release channel is just simply referred to as an RYR channel. And in order for calcium to move from the sarcoplasmic reticulum into the cytoplasm, a change in the shape, a conformational change of the DHP receptor, which is the voltage sensor, once an action potential has traveled down the T-tubule and reached this protein, opens what's called, sometimes referred to as a plug of the RYR channel, allowing the flow of calcium into the muscle cell cytoplasm from the sarcoplasmic reticulum. And on this illustration, we see the relationship between the DHP receptor, which is the voltage sensor, as you can see here in this illustration. Here we see the extracellular space, which is the T-tubule, the cell membrane, also referred to as the T-tubule membrane, and the DHP is connected to the RYR channel, and the SR membrane, as you can see, has a very similar structure to that of the muscle cell membrane, and the white indicates the presence of cytosol, or the cytoplasm of the cell, and the SR lumen in blue represents that interior storage space where calcium is housed. And so by highlighting these main areas, we know that the calcium, which is maintained in a resting state in very low concentrations in the cytoplasm, higher concentrations in the extracellular space, and higher concentrations inside the SR lumen. And when the action potential, which was originally generated at the synaptic connection, travels down the membrane and causes a change in the electrical state across the membrane, that electrical reversal will be the trigger that will cause the DHP receptor to change shape. That will, because it's connected to the RYR channel, that will cause the RYR channel to also change shape. And when it does, the stored calcium inside the SR lumen will escape and start to disperse quickly throughout the cytoplasm. And that is the connection between the excitation and the contraction. Recall that when we talk about excitation, that refers to action potential activities. Contraction is a mechanical event that is tied to the release of calcium from the sarcoplasmic reticulum, which can happen when that action potential shows up and is sensed by the DHP receptor, which is the voltage sensor. And the DHP receptor is the dihydropyridine, specifically protein, that is that voltage sensor, and is made up of 5 different units, termed alpha 1, alpha 2, beta, delta, and gamma, where alpha 1 is a relatively large portion that serves as the voltage sensor, and the conformational state, or the shape of the A1 alpha 1 component, is sensitive to any change in the electric field that occurs when an action potential shows up. And this is an interesting drawing, originally published in 2000, which shows this structure of the protein based upon knowledge of its amino acid composition. The important aspect to this is the link with the RYR channel, which is called the ryanodine channel. And the ryanodine channel is a very fast channel in that when the sensor detects the presence of an action potential, will open this channel very fast, and it's one of the largest known channels in human physiology. It's made up of 4 subunits. Each subunit is made up of over 5,000 amino acids, so making this a very large protein that exists in the membrane of the embedded within the sarcoplasmic reticulum. There were 3 isoforms of this channel. The one that we will focus on is RYR1, which is found predominantly in skeletal muscle. The RYR2, which is very similar in structure, very similar in function, is found embedded within cardiac muscle cells. And RYR3 was first discovered in neurons in the brain and is actually found in all cells. But our focus here will be on the RYR1 channel. A condition that afflicts some individuals is referred to as malignant hyperthermia. And that's a condition where a person is sensitive to general anesthesia during surgical procedures. And when a person that is afflicted with malignant hyperthermia, general anesthesia will cause an uncontrolled opening and activation of the RYR channels, causing an increase in whole body metabolism, and that will cause a person's core body temperature to increase. And as a result, many anesthetic drugs for surgical procedures have been designed so as to not activate the RYR channel. Stimulators of the RYR channel include calcium, which is normally held in low concentration and close by the sarcoplasmic reticulum. Caffeine is also used as an activator of the RYR channel. Recall, stimulators of the RYR channel will cause it to open and will lead to muscle contraction. However, the caffeine that is used, that is an activator of the RYR channel, is used experimentally with isolated muscle preparations, and is used in much greater concentrations than what is available to humans. In terms of inhibitors, calmodulin is a natural inhibitor of the RYR channel, and when it binds to the RYR channel, it will keep it closed. And calmodulin is an important regulator so that the RYR channel does not stay open for long periods of time. Lactate is another inhibitor, and lactate is a negatively charged molecule that is the conjugate base of lactic acid. And so one of the potential sites of muscle fatigue, particularly with high intensity exercise, is the excessive accumulation of lactic acid that will dissociate protons and lactate that would make the opening of the calcium release channels, the RYR channels, much more difficult. In this study published in 2012, we see a very unique reconstruction of the ryanodine channel, and it also referred to as the ryanodine receptor. And the ryanodine protein, very large protein, has what's called a cytoplasmic component, which has dimensions of approximately 270 by 270 by 100 angstroms, making this a very large channel. The TA, or rather the TM, refers to the transmembrane portion of not the cell membrane of the sarcoplasmic reticulum membrane. And the TA component of the entire protein is referred to as the transmembrane assembly. And it's the movement of the slight change in shape of the entire protein is what allows calcium to escape. So even though the transmembrane assembly is much smaller than the cytoplasmic component, we see that as a protein it is still quite large with these particular dimensions. A subsequent study not only sequenced and reconstructed the RYR channel, as you can see here, but also analyzed the mechanism through which the channel opens up when triggered by the voltage sensor. And so the channel, as similar to other channels, will transition from a closed state, which is what exists under a resting condition, to an open state, which sets up the contraction of a muscle cell. And so the magnitude of movements, as you can see here, in terms of the cytoplasmic components, varies depending upon the direction. And so here we see a shape that causes movement on the order of 4, 5, and 8 angstroms in distance. Key components of the membrane are referred to as the middle part, which is the central rim, and the four corners, which are called the clamps, and handles connect the four corners of the large cytoplasmic component. And so when triggered by the dihydropyridine voltage sensor, the corners will undergo a conformational change that will lead to opening of the channels, and the opening will increase the space of various cavities, as you can see slightly illustrated with these white spots here, that will cause that opening and allow the escape of calcium into the cytoplasm. Moving on to another very important protein is SERCA. And SERCA is another protein that is found embedded within the membrane of the sarcoplasmic reticulum. And it stands for sarcoendoplasmic reticulum calcium ATPase. And where the ryanodine channel allows the downhill movement of calcium where it's in higher concentrations in the SR, SERCA will take calcium and move it into the opposite direction, which is uphill. Because calcium has to move uphill from the cytoplasm into the larger concentration inside the sarcoplasmic reticulum, it requires an energy to do that. And so what we see here, the C refers to the cytoplasm, M refers to the interior part of the sarcoplasmic reticulum membrane, and L refers to the interior lumen where the calcium is stored. And the M's that you see here refer to transmembrane helices. And so this is another relatively large protein, not as large as the RYR channel, but the alpha helice connections of the amino acid that result make this protein very conducive for embedding within the membrane of the sarcoplasmic reticulum. The SERCA exists in a number of different isoforms, and so the nature of the green amino acids, as you can see here, referred to as the C-terminus portion of the protein, will define the isoform. This particular one is what's called the 2A isoform. The actuator, or the A domain, anchors the entire protein to the membrane of the SR that will provide further stability in addition to that stability afforded by the transmembrane helices. The phosphorylation site is the site where phosphate molecules can bind, specifically in this illustration, to the 602nd amino acid in the sequence of this protein. And the P domain refers to a relatively small sequence of amino acids that will bind ATP. Remember, this is an ATPase, and the P domain represents what's called the catalytic region. That region of the large protein that will catalyze and speed up a chemical reaction. The chemical reaction is to hydrolyze an ATP molecule. And in doing so, that will allow the movement and binding of calcium to move to the interior of the sarcoplasmic reticulum. And in this illustration, this will outline two different energy states of SERCA and how it will simultaneously bind calcium and ATP to move calcium back uphill. And this is referred to as the kinetic state. And although there are a number of steps, the entire cycle operates over a few milliseconds. There is what is called the E1, or the high affinity state, where calcium ions form a strong bond with the cytoplasmic region of the SERCA protein. And then there's what's called the E2, or the low affinity state, where the connection between calcium and the protein has been weakened. And so we use the analogy of Scotch tape, which is much weaker than that of duct tape. So if using duct tape to fix something, you know that duct tape is much stronger than Scotch tape. And that's going to be an advantage when we want to move calcium uphill. Now, because this is a weaker state, this represents the rate-limiting step for calcium transport. So if using duct tape and Scotch tape to hold an object together, the strength of that connection will be only as strong as the weakest part of it, which is the Scotch tape. And so using that analogy, the low affinity state represents that rate-limiting step for moving calcium back uphill. But if we start with the E1 high affinity state, the presence of calcium in the cytoplasm, remember, calcium showed up first because the RYR channel, the ryanodine channel, allowed calcium to escape into the cytoplasm. And so because calcium is present, it will naturally flow and bind very strongly to the cytoplasmic component of SERCA. At the same time, that binding will facilitate and allow ATP, which is generated inside the cytoplasm, to bind to the catalytic domain of SERCA and undergo ATP hydrolysis and phosphorylation. That phosphorylation, because the phosphate molecule was broken off of the ATP, producing ADP, will lead to a weaker connection of the calcium ions. But because of that phosphorylation, the protein will invert and move the calcium bound on the cytoplasmic side to the luminal side. And the low energy state will make it easy for chemical dissociation to take place. And as the calcium is released, phosphate is released, H2O is added to this process, and we move back to the high affinity state as long as there is calcium present inside the cytoplasm. There are factors that affect SERCA activity. The first main factor is calcium ion concentration. And so if calcium ion concentration increases the cytoplasm, that will activate SERCA. But by decreasing cytoplasmic calcium concentration, that will reduce SERCA activity. One situation that will do this is muscle fatigue. And so with muscle fatigue, SERCA activity will decrease and the removal of calcium from the cytoplasm will not happen as quickly. And so what will also, that will lead to a reduction in the free concentration of calcium inside the sarcoplasmic reticulum, because the SERCA activity is decreased, and inside the sarcoplasmic reticulum, calcium will bind to phosphate. And if calcium is bound to phosphate, it will not leave the sarcoplasmic reticulum, and that will prevent calcium to be released with subsequent excitation, contraction, coupling, activation. In terms of ATP free energy, that refers to the ability of ATP to be hydrolyzed. However, with high intensity or maximal intensity exercise or muscle contractions, the free energy available from ATP, or the ability to break up ATP, will start to decrease. As ATP hydrolysis is very high to start with maximal or high intensity exercise, ADP and the inorganic phosphate will start to accumulate. And that is another cause of muscle fatigue, where a person just cannot produce the same level of muscle force as they did prior to fatigue onset. One final factor that will affect SERCA activity is pH. And so with the accumulation of lactic acid with high and maximal intensity muscle contractions, the release of protons from lactic acid will drive down pH, which makes the cytoplasm more acidic. And because proteins are pH sensitive, this will have the effect of reducing SERCA activity. And as long as SERCA activity decreases, the removal of calcium from the cytoplasm will start to decrease, and that will eventually decrease the release of calcium with subsequent action potential activations. So that ends this part of our muscle physiology lecture on excitation, contraction, coupling.